Coding, but you need to apply it now to particular features. Hello, Bradley.
Hello, how are you?
Okay.
How's everyone doing? Sushmanth and Kritika and Jesse and Dick and Susan.
I won't be here next week. Just saying, I'm going to be at that APS physics conference. So.
Oh yeah. Hope it's that should be fun. Last year was looked like it was pretty productive, as I recall.
Yeah. Well, I won't be able to take notes on my computer like I did, but I'll just try to catch the references.
Yeah. Jesse said, could I get an invite to the DevoWorm Slack? Well, it's the OpenWorm Slack. I don't think... Oh, okay. You have to do it through your Joe Pro account. Okay. Well, I'll give you the invite. Actually, it should go... I'll just send you the link later. Yeah. So any updates from anyone? Anyone want anything to talk about? I know Sushmanth is doing some things, I think more things with, okay.
Okay. You and I need to talk about, did you get my note this morning?
Yeah, I did.
Yeah. we got to do it online and graphically, so we have a common understanding.
All right.
Actually, Bradley, I hosted the dataset, Epic dataset on OpenWorm organization for segmenting, and I'm pretty stuck with creating a API for DevoWorm on Hugging Face. I was taking help from one guy from Hugging Face, Brad Ems. He was also contributing. Actually, 2 days back, he kept a pull request to our repo. I merged it without your permission, actually. I didn't let you know, but I merged it. Some documentation thing, he helped me out.
Okay.
And we are creating an API for DevoWorm. Like, can I share my screen and show it to you?
Yeah, go ahead.
Like this kind of thing. Like I want to create an API. I think it doesn't create. See, I mean, can you see an API here?
Yeah, I can see it.
Yeah, like if you give some input, this is a like text-based thing model. So ours is on like segmentation mode. I was just creating an API. If you give something here, like two comparisons. There's a problem with this. Okay, like this documentation kind of thing I want to create and host our models as DevoWorms. There are 3 models, right? Lineage, population, and these 2.
Yeah.
And I want to create an API. Like complete DevoWorm will be like same GitHub.
Okay.
Yeah, this documentation is cool. I think it's okay. I hosted the data set, but the documentation for this data should be added. Actually, I just give a small documentation. I don't have clear knowledge regarding this, so I kept it like somewhat empty. But we need to add this thing. I have given the documentation, whatever it is there in DevoWorm's README file. I added here, and I want to create an API and all these things here. That's it is pending. I'm in like halfway there. Maybe within a we will try to complete this work. So I mean, DevoWorms in Hugging Face, everything will be work up and running. And according to GSOC, I mean, when can we submit that draft proposal? Actually, can I submit?
You can submit it to me to look at it. You can submit it to me anytime, but I think the application period opens March 20th. Yeah, March 20th.
Okay. Yeah, if you don't mind, Mike Deb has asked my proposal also for review. I will share it to him.
Yeah,
he told like send it to you and then share it to him. So
yeah, sounds good.
That's what I'll.
Hydroponic tomato.
Oh yeah.
Remember, we still have snow on the ground.
Oh yeah. It's great. So, Kritika, how are you? Would you like to introduce yourself? Okay. Yeah, you can type in the chat if you want. Jesse says he'd like to set up hydroponics.
Okay.
So yeah. I think she's still typing. So thank you, Sushmanth, for that. That was great. I was looking at actually just before we got into the meeting, I was looking at some of the things on Hugging Face. So it looks like it's coming along. So the API, that's great. You'll have to work on the documentation a little bit, but that's yeah.
I would have completed it, but I don't have knowledge in Java. The whole API thing needs idea in Java.
Oh, it does? Okay.
Yeah, that's the problem. I was taking help from some others. Hugging Face team, actually. They are like friendly community. I have never seen like a community like that. They are helping me out. Whatever the maintainers are there for the GitHub, they are helping me out and they are telling me everything.
Well, that's great. Yeah.
Can you have a 10-minute meet after this meeting? I just need to show some things which are like kind of regarding DevoWorm. If it's possible, just a 10 minutes meeting would be nice.
Yeah, that'd be good.
Okay.
Yeah, like some time other than now, like during the week.
Okay, if it is not possible today, okay, it's okay. Like tomorrow or like that. You can just give me a date. I will just open to the meeting.
Okay. Yeah. So Kritika said, I just started with machine learning and I'm really fascinated by bioinformatics. I haven't contributed yet, but I'm really looking forward to learning about GSOC. Yeah, that's good. Yes, thanks for attending the meeting. So, please stick around and, let me know if you have any questions or you find something that's really interesting to you. Susan, how's your project going? Oh, yeah.
I've discovered a way to do my stress strain graphs, only I have to do my model in solid mechanics and not truss structures. So I'm going to go visit them at the conference, maybe every day. They're going to feel like they've been bugged.
Yeah.
That's okay. I'm going to devote my time to visiting the console booth. Really hard to get answers out of them some days.
Oh, yeah.
Yeah. So I guess that's what I'll do.
Okay, yeah.
They've got wire structures and solid mechanics, and if I can combine the two, which they advertise as being possible, I'm good. I know what I want to do. It's just the solid mechanics adds a new level of difficulty to things because you have to be able to geometrically link them together. The truss structure, you just say, I want to draw a wire from here to there, and it does it. With the solid mechanics. Oh, you have to go from this coordinate to that coordinate and actually draw it. So, anyway, I have a few questions.
Yeah. That's great.
Susan, has anyone done a study on the stability of random tensegrity structures?
There is some sort of a tensegrity modeler out there, some software, and somebody did some programming in Symlink that's related to MATLAB on tensegrity structures. I know, you want me to look those up.
Well, Bradley and I are writing a paper in which we mentioned the possibility of tensegrity structures falling into two categories, namely those that are stable and those that collapse. And we'd like to know, if you make a random tensegrity structure, what's the probability that it will not collapse?
I think all tensegrity structures will collapse if they're tweaked the wrong way. The one I was looking at, the 3-strut tensegrity that I have to model to check my models, it collapses if it's too tall and twisted in a certain direction. I think there's a collapse point somewhere in it. Yeah, okay. Well, I could try sending you a few papers.
We had a startling result. Bradley made a simulation of random Breitenberg vehicles. Okay, and the startling result is that if we say that if we define viability by the ability to move, and I guess a few other criteria, we got 37% viability from random structures.
Oh, well, that sounds like a nice system.
Yeah.
It'll live.
So the same question could be asked about tensegrity structures. If we had a criterion for them being non-collapsing, okay, then what fraction of random structures are non-collapsing?
Oh, I'll leave that one to you.
You could do the simulations.
Yeah, well, if you find some interesting structures, I could simulate them.
Well, no. We need to randomly generate them. So that's why it's related to what you're doing with COMSOL, because the program has to generate it, not by hand.
Yeah. Well, I think I'd like to give that one to MATLAB, actually.
Okay.
So I've got somebody's thesis where they did this all in Symlink, and that might work. But I'm thinking of the paper that did some iterations on tensegrity structures, and they removed the tension elements that weren't needed, and the structure gradually became more rounded.
Okay.
It was interesting.
Yeah.
I have like a stack of papers, like.
Hey, I don't. All mine are in my computer.
Well, I print out the interesting ones because I sometimes have trouble reading on the computer.
Oh.
I just
try large print.
Yeah, something like that. Well, when you print them out, sometimes it makes it worse. They've got it at 10 font. Don't make your papers at 10 font. It just drives me nuts.
Yeah, I've got a table at 5 point.
Yeah. It might as well be.
I have to magnify it to 500% to read it.
Yeah, exactly. Yeah.
Well, yeah.
Anyway,
yeah.
I'll send you a couple of references.
Okay.
That's great. So Jesse, how are you? Haven't seen you here in a while. If he's there. Yeah, there he is. Okay, I'll be on mic in a moment if I can make it work. All right. Yeah.
Kritika, what's your background? Are you a student?
I think she's her. I don't know if her mic is working, but
Oh,
she can type in.
I'm a student. I'm currently in my sophomore year, undergraduate degree. I'm doing BTech in IT and mathematical innovations right now, and I have a minor in computational biology. So I chose that because I was really interested, and the scope is really good, and it's really fascinating.
okay. Okay. Yeah, we're Bradley and I are working also on projects on origin of life and on detecting and destroying breast cancer.
Okay, that's really nice.
Yeah.
So, do you have any reference papers?
No, well, here, send me your email and I'll.
Yeah.
Yeah, that's the easiest way.
I'll just type it.
Right?
We also have a website for the group, which is here, and that has some of the publications we've done in the group meetings. And our group is about 8 years old, so and that's Kritika's email. So you might find something of interest there. It basically summarizes what we've done over that time.
And I really wanted to ask about the project which was listed
Yeah.
in the which was listed on the Neurostars.
Yeah. So let me see if I can pull that up. So this is Neurostars. Let me share my screen here. It comes up. Okay. I'll share my screen. So let me see if I can find the project. Here it is. So there was one last year, actually, but this is the 2023. So last year we did work on graph neural networks, and we kind of built a pipeline, and we built some software for using graph neural networks to extract graphical information out of microscopy images. So, microscopy images are, of course, things that allow you to see things that are very small, and there's a lot of the images have, our images are basically embryos that we're training our graph neural networks on, and those embryos have cells that have spatial organization. So that spatial organization, we can break that down into a graph structure, and then we can take that graph structure and extract it using the GNN and build embeddings that we can then use for other types of applications like machine learning. So, last year we built a pipeline to sort of flesh out how that would go from the microscopy images to the embeddings, and then worked on some of the software. But the problem, of course, was that was a first pass, and we found that it was very hard to sort of get our heads around what that should look like. So we got some progress done last year, and it's sort of in place, but it needs to be improved, and it needs to be integrated with some of the other stuff we've done with deep learning. We have a site called DevoWorm, which was what Sushmanth was working on through the Hugging Face interface. But there's also a GitHub repository for DevoWorm. Oh, there, cat. All right. I'm going to put the DevoWorm link in here so you can look at the GitHub repository. And that's in the chat. And that should give you like an idea as to what the code looks like and the different. So DevoGraph is where, that's where the graph neural network work resides, and then DevoLearn is where the deep learning work resides. And so this project this year is going to involve sort of maintaining it, moving it to the next level, and integrating the deep learning with the GNNs. So graph neural networks are a potentially powerful method, but we have to sort of get our hands around what we have. So What we want to do is develop these developmental GNNs that allow us to characterize a growing network of cells. So in embryology, cells are constantly dividing and creating the structure. And so we want to be able to take images of that process and extract the sort of the graph embeddings that exist in there. So there's some graph structure in there. We want to recover it and then build embeddings for other purposes. So we built a roadmap and a pipeline for this, but we weren't able to develop full integration, and we still need it still needs a lot of work in terms of, getting it to work well, because the stuff that we have, we train it on the data that we can find. And so if we have, there are a couple of data sets that we have that we can train the models on, but by and large, this is kind of a hard, it's a hard thing to really get to work well, because we're limited by the kinds of data we have that will work. So a lot of the data is not really optimized for image segmentation, for example. So it's hard to really kind of use that effectively. But we'll keep working on it this summer. So if you propose a project, your proposal should focus on what exactly you want to what exactly you want to do within the scope of the GNNs initiative. So it could be like, taking the existing code and improving it, or introducing maybe a new way to do this, or maybe working on the pipeline, working on the integration between graph neural networks and deep learning. There are a lot of ways that you can work on this project. I would suggest looking at the code on GitHub, and kind of thinking about maybe what you would want to do, and then I'll be available for feedback. So if you have an idea and you want to put it in I think you're in the Slack, correct, Kritika? Okay, so
Thank you so much.
yeah. So yeah, if you have a question, you can ask there, and then if you have your proposal, then you can pass it. You can send it to me, and I can give you feedback. So
Thank you.
yeah, no problem. All right. So Jesse, all right, do you want to say something, or notice you switched your account?
Yeah, I don't have a whole lot to say, but I'm just trying to get up to date, up to speed again. My schedule's worked out where I can come back to the meetings a bit more often, and I want to kind of help out with Google Summer of Code a bit if I can. But also, there's a lot of older like papers and things that were here that I haven't really touched on recently and want to kind of pick up again. So no major updates to me, other than I hope to be I should be able to actually virtually physically meet on the Monday meetings more often. So I want to just kind of do that, and it's good to see everybody again, and more in the future. But that's all for me, really. Good to see everybody.
Well, yeah, great. Thanks for coming. And I look forward to, if you want to be involved in Google Summer of Code, looking forward to that. Definitely, we'll try to do, more when we start Google Summer of Code. We'll do the community period, and then we'll do the coding. And community period will be something you can help with, definitely. And then the coding period will be just kind of like feedback on things. And so definitely, I think that'll be something that, you can also help with. Yeah. So anything else before we move on?
Bradley, do you want to? I don't know if you want to do the circles here.
Well, yeah, we could do it here. Sure. Yeah.
Oh, we could. Okay. Well, why don't you pull up what you got?
Oh, I don't know if I have it. I don't know if I have it up. Yeah. But let's see if I can find it here. Right.
Worst case, it's in your email.
No, I got it. see. I'll share my screen. Okay. There we go. So these are the images. I have figure 4, which is this summary image. What was the thing that, yeah.
Okay. The molecular world is for people who work in origin of life. That does not mean anything.
Okay.
What we mean is well, I put it in terms of chemistry language, that it's the bath in which the vesicle exists. And the bath is usually infinite compared to the vesicle.
All right.
Okay? Okay, show that one more time.
What do you mean? It should be up.
Oh, there it is. Okay.
Yeah.
Okay, now the circle is correct, but there should be a line for the circle, which is the membrane. It can be a thin line.
Around the edge.
Yeah, around the edge of the circle.
Yeah. I got it down, the way I draw this, it's like I've got it down to 1 pixel. So that's the thinnest line I can make.
Oh, I can't see anything.
Well, I mean, I made the line that I had in the last figure was the thinnest I
that
can
may
make
be
it.
the problem. It
Yeah.
has to be big enough to be visible, like the lines that you have on straight lines that are there.
Yeah.
That would be fine. Okay, show me in one where the diameter is smaller.
Like, you mean like this one here?
Yeah, that one. Okay, so
Okay.
before it's 40 micron diameter.
Yeah. Yeah.
Okay, the two outer circles should not be there,
Oh, okay.
and the inner circle should again have a line.
All right. All right. All right. Let me see if I can pull up the one with a line. I don't know if I have it in here. I think I have it here. No. Yeah, this one, I think. This one has the line here. So this is the 1 pixel line, I think.
Okay, yeah, the inner circle is wrong.
Okay.
Go all the way. It should not be there. It should go all the way out. The gray should be all the way out to the edge.
All right.
The outer circle.
Yeah, yeah. Like,
no inner circle.
all right. And then this outer edge, yeah, this is like, if this is 1 pixel, how do I make it? I don't know how to make it any smaller than that or thinner.
Oh no, that's fine. That's fine. It's not. It's just supposed to be thin.
Oh, okay. All right, I see.
Okay, right now you have it thick because of the inner circle.
Oh, okay.
Okay, so the inner circle should be expanded to the outer circle and disappear.
All right.
Okay, and the other thing I didn't understand is if we set K, which is the number of kinds of molecules, to 36, how can you ever get something bigger than 36 for any of the simulations?
In terms of kinds?
Yeah, in terms of kinds.
I'm not sure.
Okay, that's the other confusing part.
Oh, okay, I see what you mean.
Okay. It seems as if you have that many kinds, you can't go any bigger. So the fluctuations should go up to that, but not beyond it.
Right.
Okay.
I'll have to check. I know there's something that there's an answer to that. I don't know what it is.
There's something something incongruous there. Okay. For Kritika, the reason I'm jumping, I seem to be going up and down because I'm on a treadmill.
Okay.
It's a treadmill with a desk, and the desk has my computer on. Okay.
Yeah. Okay, I think that's great. I'll address those questions and get back the revised figures. Yeah.
Right. Okay. Thanks. Okay.
Jesse said, I'm on a treadmill too. It's the publication cycle.
Yeah. That's a figurative one. I'm on a real trip. Are you tried taking a walk yesterday, and the wind, even though it's not very cold, the wind is bitter.
Yeah.
So it's still indoor exercises.
I'm escaping. I'm going south.
You're going south. Oh, okay.
Yeah. Well, next week.
Okay, bring back some of that global warming.
All right. So I'm gonna move to some things here. Let's see. I got a couple of articles that are kind of long, and I'll just go over them a little bit. But they might find them interesting. I'll post them later, or send them to you if you find them interesting. So the first one is the Stephen Wolfram article, and he has a blog that is very prolific on a lot of things. So he just published something on ChatGPT. So but he has a series on the Second Law of Thermodynamics, and this is like a 3. I think it's like he's tried. First of all, I think he's tried to solve the second law. So I don't know how effective the solution is, but he does have some interesting things to say about thermodynamics in terms of its history, and so.
Bradley, could you please put a URL in the chat?
Yeah, let me see. There you go.
Thank you.
Yeah. So this is kind of goes over like the origins of the second law. So the basic concept of the second law was first formulated in the 1850s and rather rapidly took on something close to its modern form. It began partly as an empirical law and partly as something abstractly constructed on the basis of the idea of molecules that nobody at the time knew for sure existed. So this was in the 1850s. This was pretty early on in the history of this. So this was something that emerged from a lot of things going on with the study of mechanics and heat and things like that. But by the end of the 1800s, with the existence of molecules increasingly firmly established, the second law began to often be treated as an almost mathematically proven necessary law of physics. There were still mathematical loose ends, as well as issues such as its application to living systems and to systems involving gravity. So it was, this is like the standard model of the second law is this closed system where you have a closed container and you set it to equilibrium and then see how long, entropy takes to bring it down to near zero energy. So a lot of the models of the second law, suggest that you can't create nor destroy energy. You either have it in this active state or this entropic state. And there's this decrease in energy, or there's this increase in entropy over time that eventually you get the system being mostly entropic, and then that runs down into decay, but that's a closed system. So usually the way they measure it is just to close the system off to new energetic inputs and measure the decay. There, of course, are open systems, which is most of life and some other, a lot of other things, but that's with an open system, it's a different, a different scenario. So this is kind of the standard thing that we have. But the almost universal conventional wisdom that became the second law must always hold. And if it didn't seem to in a particular case, then that just must be because there's something one didn't yet understand about the case. I don't know. I guess it's just that it applies to everything. And if it doesn't quite match up, then there's something you're not understanding about the second law. So the second law can never be failed. So this is, I think, maybe motivating some of his work on this. There is also a sense that regardless of its foundations, the second law was successfully used in practice. So, there are a lot of application domains for the second law in chemistry and engineering, for example, and you do a lot of computations of entropy, and those turn out to be correct. So there can't be any real problem with it because it's consistent with empirical observations. But despite its ubiquitous appearance in textbooks, when it comes to foundational questions, there's always been a certain air of mystery around the second law. So this is something that he's I think he's exploring in some of these other posts in this series. Though after 150 years, it's typically an assumption that somehow it must have all been worked out. And so this is kind of motivating some of his work, but it gets into some things about the history of heat, which sounds a little strange, but like it's actually quite fascinating. And it kind of goes into some of the early concepts of heat and how we've really kind of worked from the concept of heat to the concept of energy, and then heat energy, and then that allowed us to discover the second law. So in seeing fire, one might imagine it as a disembodied form of heat. In ancient Greek times, they talked about everything somehow being made of fire and also somehow being intrinsically in motion. So these are metaphors that were around a long time ago before they knew anything about molecules or the second law. So this kind of is built upon our understanding of these processes, and it kind of goes on and talks about the more recent history of sort of the things leading up to the second law. So last week we talked about what was the topic? Commensurability in the philosophy of science. And commensurability is this thing where we have to sort of have, the ideas in our science sort of formulate how we come up with theories. So our concepts sort of lead to our theories. And that's kind of what he's talking about here, where some of these concepts of like what heat energy is and how it works kind of inform how we came to arrive at our current conception of the second law. So it's really interesting, kind of some of this early history of science leading up to the second law. And of course, it's Boltzmann who proposed the second law. And he's in the physicist in the 19th century. So really the beginning of thermodynamics itself started with heat engines. And so the Industrial Revolution started off, and one of the sort of the key innovations there was the steam engine, or efficient steam engines. And so there is a practical concern there as to build sort of this optimal steam engine. And to do that, you have to understand things about heat energy, and you have to understand things about entropy. So, it goes through James Watt, Carnot, and, some of these things that they were doing with machines and became statistical mechanics. They didn't know it yet, but that's where this is all going. And then there was this work on, simultaneous to that, there was this work on caloric theory, which was not correct as a conceptual framework, but this idea of reversibility and operating in a closed cycle, those things were correct. And so they ended up contributing to the second law. So actually Carnot introduced the second law, but he was ignored. And then the idea came up again. So people were converging on this idea as it was being devised. So that's, James Clerk Maxwell actually was the more sort of famous person to come up with these ideas and put them together in some of the work that led us to the second law. So this is a really long article. I don't want to go through the whole thing, but any thoughts on that? They didn't talk anything about biology, though, really. This is mechanics. But, you can see that in biology, it's been applied quite extensively in terms of like energy consumption and in terms of the reversibility of energy and things like that.
Does he say anything about the status of entropy in non-equilibrium systems?
I don't think in this article. Well, let's see. Go to the end here. It's a long article. I don't think in this article, though. No.
I think this is what Kritika got the Nobel Prize for. It's extending these concepts to non-equilibrium, wakes, in liquids and stuff like that.
Yeah. Yeah. Susan, do you have any thoughts? I know you're...
There was a fellow who tried to apply maximum entropy to non-equilibrium systems. And I can recall as a graduate student doing a precise calculation of a non-equilibrium system and his time calculation, and his didn't work.
Oh. Yeah.
Jane, I think, Jayne.
Okay.
The stuff may still be around.
All right.
He'll be 60 years old.
Yeah. Taking some notes. Oh, Susan, did you have any thoughts on this since you're doing a lot of mechanics?
Systems, you have to be careful with it because we're basically a chaotic system. And if you tie it down to, say, Boltzmann's constant, then you're not looking at the full behavior or something. But that's all kind of off to the side, I guess, of what you're talking about. But yeah, it's used a lot in seeing if a system is stable and so on. I'm not a fan of necessarily the second law. Yeah, so, oh, well, what defies entropy? And I say life does.
Yeah, yeah.
And I told that to a physics teacher one time, and he says, I'm not touching that with a 10-foot pole. He'd have the entire school in an uproar. Went with that. But anyway, yeah, this is sort of, and it's an interesting topic.
Yeah. Yeah. So, well, I think that's the part that we didn't talk about here, was that life is an open system instead of a closed system. And so it's not so much that it violates the second law, but it's definitely like, you know, we need to think about it a little bit differently when we think about life. Because there are always influxes of energy coming in. And of course, you get structure built from entropy, but you also get structure built from flows of energy coming in. So, and there were a lot of things like dissipative structures and other types of concepts that came a little bit later that kind of get in touch on that. So, yeah. So the other thing I wanted to talk about here too was this from this blog, Molecular Tinkering, and that's this this thing about the epigenetic landscape. So this is piece. I'm really trying to make the most of the epigenetic landscape. So this is a picture of a mountainous landscape, a rugged landscape. And they talk about development here, and they talk about Conrad-Waddington. And for people who aren't familiar with Conrad-Waddington's work, he's a developmental biologist who developed he wrote a number of books back in the mid 20th century, and he wrote a book, at least one book, on this idea of the epigenetic landscape, which they call the Waddington landscape. The idea is that it looks like this surface that is undulating, and that you start at the top, and you have a ball. It's usually a ball, and it rolls down the landscape, and it rolls into these valleys, what they call canals or canalization, which is where the ball rolls through the area of least resistance, which are these valleys, and then down to the bottom. And these valleys actually branch in a tree-like way, so that you end up with multiple possible endpoints in your tree. So down this, you go down the landscape, it sort of forms this set of, it's almost like a river, where, kind of like a reverse river system.
Bradley, did you ever come across a definition of the ball?
No. No, that's yeah.
What is the ball?
Yeah. So the problem is yeah, it's highly something that's highly like abstract. The ball is supposed to be developmental potential, but it's kind of like it's not. That's not the greatest analogy. So so it's very vague. But that's a lot of in biology. We have a lot of conceptual models like that, which is maybe good and bad because
Yeah.
right.
Oh, by the way, I postdoced with them.
Oh yeah. Great.
Okay.
Yeah. So Waddington was the last Renaissance biologist, and he had a background in philosophy, paleontology, embryology, and genetics, and he was what they call a polymath, which means he had a lot of different specialties in one place. And so he one of Waddington's most enduring gifts to biology is not a fact, hard won through an elegant series of experiments. Instead, it was an analogy derived from an understanding of the data available to him, an intuitive notion of how embryonic development should proceed. And so he actually talked about the key to understanding the building of an embryo is to see it as a gradual process of specialization. Through its life, each cell undergoes a series of choices. With time, the choices become more specific, most irreversible. So he talks about this irreversibility. This is kind of like an entropy when you get this irreversible process of entropy and the need for more energy to be supplied to move it forward. So to visualize this process, he came up with this hypothetical landscape. The cells of the early embryo can be thought of as balls at a high point in a craggy or eroded landscape. And as the balls, which are these analogies for developmental potential, roll downhill, they will enter a system of branching valleys. There are many possible routes down the hill. Each fork in the valley system represents a decision point for the cell. So that, there are different decision points in development. Some of them are sort of the differentiation of two things. So endoderm versus ectoderm, foregut versus hindgut, pancreas versus liver. So as differentiation occurs in the embryo, which it starts out as this undifferentiated mass of cells, you get these points where things get differentiated, and those are these sort of branching points in these canals. So the ball can go down one way or another. So this is a visually appealing metaphor, but then of course we don't know. I mean, that's just a metaphor. Waddington then wrote a book, The Strategy of the Genes, where he laid out the sort of more detail about the underside of this model, where you have these they almost look like strings attached in different ways to the bottom of this model. And these are actually, this is a view of gene function. So you have these genes that are being expressed combinatorially, and they're underpinning these canals. So they're basically instructions to the structure of this canal, and then the ball itself is rolling in one direction or another. So this is how he kind of, this is really kind of pre-genome, pre-human genome especially, and this is still analogous to gene expression and sort of a view of gene function, but it's actually not very specific in terms of what we know today. So, but it's still interesting because you can get the basic structure of gene function is correct, I think, in that there are a lot of interactions. It's just the way, putting like specific things on these boxes. So these black things are genes, these black lines are, different ways that they can be expressed, and then they underpin these canals. So it's all very, it's all a bunch of analogies, but that's what you need in theory to really have a successful theory. Is you need a very generalizable model. So,
okay, might I add to this, the differentiation tree is essentially an embodiment of Waddington's landscape.
Yeah.
Okay.
Yeah.
Which needs a lot more testing. It might work.
Yeah. Yeah. And then Susan put something in the chat here. So she had this citation here from 2020 modeling and simulation of tensegrity structure based on sim mechanics. So this must be the one on the random tensegrity networks or tensegrity structures.
No, that's just modeling in Simlink. So that's MATLAB.
Yeah.
Okay.
Yeah, and then the reference for the triangular tensegrity is in my PowerPoint that I presented and sent. So yeah, that was, yeah.
I showed it to you. That's the one where they had elastic strings and then stiffer strings, and the stiffer string produced the J curve that's more like a cell produces.
Yeah.
Anyway, this there's a form finding method for tensegrity structure in the thesis here, the modeling simulation with the sim mechanics.
Yeah.
Of course, I
Printed it out.
Put it in this. Yeah, so they have a form finding method in this thesis, if you're interested.
Yeah.
And they've got the three strut or triangular tensegrity in here too. So yeah, they just kind of interesting. They actually built a physical model and put a motor on it, and it's interesting.
They put a motor on it.
Yeah.
So it moves?
They have a motor. They have a tensegrity, and then they put a motor.
Wow.
Motor on it.
Okay.
Like, okay.
Tensegrity robots.
Well, I don't know. What does that look like?
Well,
doesn't look like the NASA robot.
No, it's different. I think looks like they're moving it around or something. They think tensegrity structures might be good for earthquakes.
Yeah.
Oh.
I mean, super stable structures.
Well,
Yeah.
they're stable when things are going
Right.
wrong.
Right.
So.
Well, yeah, that brings up an interesting question. Like, how stable are they in motion? Like, if you have something moving through a medium of like water or, air or something like that, it's like we're testing it. I guess we're testing it statically, maybe dynamically. We're not testing it like in a environment where an organism might move. Yeah. Imagine that's
Stephen Levin thinks all living things are tensegrity structures.
right.
Fractal tensegrity structures.
Yeah.
Susan Levin twisting his arm. We can't get him to write a book.
Right. He's answered my email, so I keep sending him. I sent him my presentation, and he liked it. And he says, "Yeah, but you're forgetting this, that, and the other thing." I'm going, "Well, no, I just didn't include them. Like it was a 20-minute speech." Anyway, it's okay. He'll, he can give me clues. It helps actually, just to keep them in mind. Like if somebody asks, "Well, what about the sides of the cell? Aren't they contributing to things?" Well, he says there's, hydrolonic acid in there that's very slippery, that has no friction. So really, the cells themselves control their attachments. They're in the actin cadherin attachments and integrin attachments. The cells actually control this, and then they control their own environment by creating the basement membrane or the extracellular matrix. So they're in control of their environment to quite a great extent.
Okay.
Yeah.
Yeah.
So
I gotta go. I got a webinar coming up.
Okay.
Oh,
yeah.
All right.
Thank you. Bye.
Bye.
Okay. All right. I think that's enough for today. If anyone else had anything wanted to say, I can say it now. Otherwise, Okay, now I'd like to talk about something that we published in 2021, and then I want to talk about like a follow-up to that in a recent paper. And it's this concept of developmental tempo. And so let's go to the first of all, this is the paper we published in 2021, and it was published in Biosystems. And the title of it was Periodicity in the Embryo, Emergence of Order in Space, Diffusion of Order in Time. And this is Ujwal Singh and Jesse Parent, as well as myself. These are all collaborators in DevoWorm. So the abstract reads, does embryonic development exhibit characteristic temporal features? This is, of course, apparent in evolution, and this was something that Stephen Jay Gould talked about with respect to the tempo of evolution, or the tempo of phylogeny. And so this means that change occurs in bursts of activity, where the change occurs at a certain sort of cadence across time. And so what we're trying to do here, instead of looking at tempo in evolution, we're looking at tempo in development. And so it's a very similar concept, but not precisely the same concept. I don't want us to be criticized for saying that, we're looking at evolutionary tempo sensu Gould when we're actually not doing that. But in any case, what we do in this paper is use two animal models, the nematode, which we talk about in the group all the time, and the zebrafish, and that's another model organism. And it's a very different organism in terms of its development. We also use simulated data, which is a numeric simulation of sort of this tempo idea, taking the idea and applying it to the branching structure of a lineage tree. So we use those three sources of data. In doing so, we demonstrate the temporal heterogeneity exists in embryogenesis at the cellular level and may have functional consequences. Cell proliferation and division from cell tracking data, which is used for zebrafish and nematode, is subject to analysis to characterize specific features in each model species. Simulated data is then used to understand what role this variation might play in producing phenotypic variation in the adult phenotype. This goes beyond a molecular characterization of developmental regulation to provide a quantitative result at the phenotypic scale of complexity. So this is a comparative paper that uses, of course, data from C. elegans, data from Danio ririo, which is the zebrafish, and a numeric embryo, which we have created. So the C. elegans data and the zebrafish data are both things that we acquired from the System Science of Biology database. And so this is based on cell tracking data for C. elegans, the Bao et al. Paper in 2006, which is basically the precursor to the EPIC data set. But this is data where all the cells are tracked in development in C. elegans. And then the Danio ririo or zebrafish data set is based on the cell tracks of nuclei and Keller et al. 2008. So they created a data set that's a standard for zebrafish cell position and development. The cell tracking data is used to determine the total number of new cells, or the cell division time, present at a particular time step. So we're able to characterize sort of the time course of development in each organism, and then look at those independently to see what those look like, and if we can extract common features from those two data, from those two time series. We also used a numeric embryo, which was where we generated pseudo data based on a lineage tree, which we would be able to characterize in C. elegans and zebrafish by cell divisions. We do a similar thing here. We generate pseudo data, and each numeric embryo consists of one or more vectors describing rounds of cell division, and then we can use that to build a graph to show compare it to biological data, but also play around with it in different ways to determine the tempo of development and what that looks like in terms of cell divisions. So the results here are where we compare C. elegans with zebrafish. It's obviously a very different type of different tempo of development. In C. elegans, its development is completed well before 1,000 minutes of developmental time. In zebrafish, we not only have a longer developmental period, but we have longer rounds or longer bouts of cell division. And this is just because it's a more complex phenotype, and it gives you this pattern where you get this initial burst of cell division, which is a little bit off center from what you see in C. elegans, but it's very crowded with cells. And then these are histograms, so they show the frequency of cell division at a certain point in time, but you also have these rounds of cell division later in development between 1,200 and 2,000 minutes and between 2,400 and 3,000 minutes. So this just points to some of the differences in the embryo and the biological complexity of the organisms, but you do see some commonalities as well. And so this is the interval between cell division events and embryonic development. You can see that there's a large number of events that happen rather quickly. So you have cell division events that are rapid, and then you have less rapid cell division events. There are a couple outliers here where it takes almost the entire duration of development for this particular cell to divide. And this is what you see in the lineage tree. But stating it in this way is a little bit more clear as to the sort of distribution of speed of this process. And you see the same thing here this is, I think, for zebrafish. So this is where you get, this red window is, this is a periodic region and an aperiodic region. So basically this is for up to the gastrula stage in development. And so this is a total number of cells, and it goes out to like 3,500 cells. And the number of new cells, of course, looks like they're these distinct rounds of cell division up till about 1,500 minutes, or up to about 1,500 cells, I'm sorry. And then after the 1,500 cell stage, then you get these more aperiodic division events that have, some structure. We look at structure, we look at like kind of the unitary nature of these bursts. So this is kind of a, without using like high-end machine learning, we can segment this time series in this way. And so we draw a box roughly around this periodic set of stages of cell bursts. So this is just a statement to show that there's this transition between these periodic bursts to these more aperiodic bursts of cell division and the number of new cells that occur at each interval of development. So you usually have, when you say something goes from an 8 to a 16 cell stage, the cells double, all the cells divide, but in some of these embryos, you're going to have asymmetric division events. And you'll tend to have more asymmetric division events in the aperiodic stage. So looking at it in this way is a little bit different than just saying that something is symmetric versus asymmetric, or that, classifying the embryo in different stages. It's just a different way to describe it. This talks about this graph is the relative frequency of division rate across developmental time in zebrafish. So this is in zebrafish, looking at the frequency of division rate. This is analogous to this other graph that we showed before for C. elegans. This one here, it's just a different way because we have to do it a little bit differently for zebrafish for a number of reasons. But basically, we have a similar situation where we have cells that divide quickly. A large number of the cells divide rather rapidly. So you have these cell division events that are rapid, that maybe go over 2 or 3 minutes. And then you have a few cells that actually undergo a much longer period in between their division times. So we go from the mother cell to the daughter cell, and how many minutes, or what's the sampling interval. So we get this distribution, and it's similar to C. elegans. And then we look at what we do a lot of work on embryo networks, which are extracting networks out of embryo tracking data. So we look at the distance between the cells, we look at the position of the cells, and we can build these spatial networks that are called embryo networks. So these embryo networks, we characterize them for zebrafish, and this is figure 6. So if we go down to, this is figure 5, this is just the interval size of peaks in cell division for all developmental cells. This is, again, another transformation of the original data. So the zebrafish is actually notable, and that's very different from C. elegans. C. elegans forms this sort of mean, this normal distribution with a mean at about 4. It's a little bit rough in terms of a normal distribution, but we'll take it. Whereas the zebrafish, you have the multimodal distribution in terms of the interval size, in terms of minutes. So there's a lot more diversity in terms of the timing of zebrafish embryos. And that leads us to this sort of network here, where we have this embryo network at the 239 cell stage. And the way this works is this is not like in any sort of space of the embryo. This is showing the interactions between cells, just to show that if there are more connections, you'll see more lines. So it's more like spaghetti if there are more connections between cells. What you see here is local clustering, so that makes sense because we know that the cells are segregating, and we think that these bursts or these differences in interval, and also the bursts in one of the previous graphs relate to this sort of structure in the graph. So you have these clusters, these local clusters, which have their signatures in the timing of division. And you can see that in the embryo network. You can also see a three-dimensional representation. So this is an embryo network that is spatially discrete or spatially explicit. And you see that these blue and red and black dots all have, let's see. So this is cells in developmental location color-coded by status in the network. The white cells are all cells not above the threshold. So we threshold our potential connections between cells, and they have to be within a certain threshold, a distance threshold, to be to be actually considered to be connected. So all white cells in here are not connected to this network. The network, you can see, kind of clusters in the middle of the space, which is expected. The red cells also are cells with at least one edge to another cell. So the red cells are connected at least once to some other cell. The blue cells are all destination cells with at least one edge to another cell. So the red cells are source cells, the blue cells are destination cells, which means that this is a directed network. The red cells are providing a connection, or, in terms of time, the blue cells are destination cells with at least one edge to another cell. And then black is all cells with more than 8 edges to other cells. So the black ones are really the ones to pay attention to. These are the hubs in this network. And red and blue cells are basically just the peripheral nodes. But you can see the black cells are clustered in a certain spatial area. These are the hub cells. These are the ones that are in the middle. And these are the ones that are organizing a lot of the structure. Okay. So then finally we get into the numeric simulations. This is where we have the number of divisions versus developmental time. So we have this first, this blue function, which is, I think, a random, this is a uniform distribution. So the uniform distribution is in blue, and it's like a staircase. It just goes up at regular intervals. And this is just a cell, a lineage tree that divides at, regular intervals, and that's deterministic. So we end up with this staircase. By contrast, we want to look at other things. We're not really interested in the normal distribution, the standard case. We want to see some variation. We want to see lags and things that shoot ahead and so forth. So we actually have modeled this with other types of distributions, an exponential distribution in orange, which shows a sort of staircase. The Poisson, which is gray, that's just another kind of version of exponential. It's not really exponential, but and then binomial, which is yellow, and that's another type of distribution. And you can see those different distributions pull away from this normal distribution. And so we can actually think about this as like these other types of distributions as the biological case, as opposed to the normally distributed case. The normally distributed case would be sort of like the null hypothesis, and the others would be sort of ahead or behind at certain points in development, which is something we would expect to see from a biological context. And for the reasons that we showed, there's diversity between organisms. There are different processes that are spatially explicit, that are temporally explicit, that are playing a role here. And then I think that's it. So that's the whole study. We kind of talk about these simulated these simulations. We talk about them in terms of organismal outcomes. So we didn't plot this against any organismal systems, but we did plot this sort of as a way to show. And I mean, you can look at the graphs in the previous, the results in the previous graphs and understand that these alternate distributions are sort of a better fit for the data. And so that's all we had for that. That's basically the idea of tempo and development and how this might work in a whole organism. And then also the sort of the computational underpinnings or the numeric underpinnings of that. So this is a paper I wanted to get to. This is a more recent paper. A stem cell zoo uncovers intracellular signaling of developmental tempo across mammals. And so our embryo networks actually do focus a bit on intracellular signaling, because the idea is that you have these areas that you have the distance between cells sort of governs the intracellular signaling that occurs, and that sets developmental tempo, but it also sets spatial interactions. And so there are a lot of interesting things going on there. So the abstract reads, differential speeds in biochemical reactions have been proposed to be responsible for the differences in the developmental tempo between mice and humans. So now they're looking at mice and humans and the difference there. And so people have looked at developmental tempo in mammals, and they've looked at this sort of idea of cell division and biochemical reactions. But they're not taking a computational view here. They're taking a biochemical and a mechanistic view. However, the underlying mechanism controlling the species-specific kinetics remains to be determined. So now they're going to be looking at species-specific kinetics. And so this is going to be a study of pluripotent stem cells. And they're going to be looking at segmentation clocks of diverse mammalian species. Segmentation clocks in diverse mammalian species varying in body weight and taxa. So that includes marmosets, rabbit, cattle, rhinoceros. Together with the mouse and human, the segmentation clock periods of the 6 species did not scale with animal body weight, but rather were grouped according to phylogeny. So what that means is that, if we back up a little bit and think about like what scaling is generally in biology, or in this type of biology, is that you have scaling that occurs according to body size, body weight, and then metabolism might have a linear relationship with body size or body weight. So we expect that things like timing for development might be tied to the size or the weight of the organism. So for smaller organisms, for example, they have a higher metabolism than larger organisms. And so we expect this for the segmentation clock. But we don't actually see this with animal body weight. We see this with respect to phylogeny, suggesting that there's a common ancestry for certain tempos. The biochemical kinetics of the core clock gene, HES7, which is a clock mechanism. So cell division is regulated by this sort of clock mechanism, or this timekeeping mechanism. It's a circuit that regulates the clock of cell division. So there are a number of stages of cell division that have to happen in sequence, and sometimes they get slowed down, sometimes they're get sped up. So that changes cell division timing. And when that changes, you get the changes that we saw in the last paper. So this gene is actually responsible for a lot of this. However, the cellular metabolic rates did not show an evident correlation. Instead, genes involving biochemical reactions showed an expression pattern that scales with the segmentation clock period. So this means that expression patterns for certain genes scales with the segmentation clock period, meaning that there's this relationship between gene expression in this period and then the developmental tempo. Altogether, our stem cell zoo uncover general scaling laws governing species-specific developmental tempo. So if we go back to scaling laws, we know that scaling laws are generally a bivariate relationship. So you draw a bivariate graph, and it's usually like one variable over another. So this could be like body length, and this could be like some fact, or I guess y, because it's actually the y-axis. And so this would scale. So for different body lengths, you get different values of y. And this is actually, it's not usually linear, it's usually log linear. Usually you get like a logarithmic function where things kind of tail off at the upper end of body length. But it's usually controlled by one variable over, so like for different values of body length, you get different values of this x variable. And so, for the clock, you might have for metabolism, for example, y might be metabolic rate, and this might be body length. And you can basically say for different body lengths, you have a different metabolic rate. There could be you could say tempo. I mean, you have to define these variables. That's why I use y here. But for tempo, you might have like a longer tempo or higher rate of change or a lower rate of change, depending on how you set this graph up with respect to body length. And what they find actually in this paper is that it's actually a matter of phylogeny and not body length, which means that phylogeny, of course, is very different. You have this tree, and you have common ancestors, and those common ancestors acquire a certain tempo. All right, and then those things are conserved and passed on to the descendant species. So all these species are going to have the same or similar tempo. And if you think about this in terms of like a cell division clock, you would acquire certain genes and they'd be in a circuit, and they'd have this certain tempo of cell division. So we actually want to look at this one. And then that's conserved across the descendants of the common ancestor with some variation. So these genes in the circuit might exhibit mutations in different descendant organisms in the tree, and they might have different rates of change. So the rates of change might be variable here, but the circuit will remain intact for the most part. And for example, but also in this clade, you might have a different set of genes that result in a different tempo. And again, you may have this local variation within the circuit. But this is how they sort of hypothesize that the zoo works. So for these type of cells that they're looking at, pluripotent cells, they can look across species and make reasonable predictions. So this paper kind of focuses in on stem cell and pluripotent cell technologies. They're using pluripotent stem cells from diverse mammalian species. They're actually looking at embryonic stem cells and induced pluripotent cells. But this is, of course, these are cells that are immortal, meaning they can divide infinitely. These are cells that they have this, we think about stem cells as being sort of, what they call pluripotent, which means that they have the potential to take on many fates. But of course, there are a lot of different types of stem cells. So that pluripotency is limited to maybe like any kind of embryonic cell or any, neural stem cells or any kind of neural cell. So it's not exactly any kind of cell. And so in the case of stem cells, cell divisions are rather important, as we saw in the developmental case in zebrafish and C. elegans. You have this, those are all developmental cells or different forms of pluripotent cell. So these aren't cells that have differentiated, they're sort of on their way to differentiation. So this is a good way to look at cell division and sort of this tempo. And of course, the tempo of development leads to all sorts of different structures, all sorts of different diverse phenotypes. One of the benefits of being able to play around with this tempo in development is to end up with different types of specialized structures. For example, if you have to grow a brain much larger than you would normally have it in that lineage, there might be some modification to the tempo of development rather than the actual cells themselves. And that will just lead you to having more division, rounds of division, and a larger structure overall in a certain part of the body. And so that's why we're interested in this. This paper is, it kind of goes through this idea in stem cells and different species. They've kind of come up with this neat, unique experimental platform, and they've been able to actually model this using the segmentation clock. So I think there's more to say about that, but not right now. I hope you learned something. We'll wrap it up.
Bradly,
Yeah.
Can we have the meeting actually, like 10 minutes when I ask before?
Yeah, we can do that right after the end of this. So.
Okay.
All right. All right. Well, thanks for attending. If you have any questions about anything, GSOC or anything related, let me know. And I'll be around in Slack and email and all that. So have a good week. Thank you.
Thank you. Bye.
Bye.
You in two or 3 weeks or something. 2 weeks.
Yeah. 2 weeks.
Yeah.
Well, have a good week next week. Yeah.
Okay.
Bye.
All right.